here and everybody doing this, and you're trying to convey something you're really interested about. So our first speaker today is Macarena Martin. Many of you know her. She asked me to remind you she's not from around these parts. So Macarena joins us from Spain, and she came to King to be a part of the swim team. On the swim team, she has been one of the captains, and she's been a standout swimmer. In the classroom, she's also a standout. She's a double major, biochemistry and mathematics, because she's crazy like that. And added the math major on this time last year. So in addition to being an overachiever in the classroom, she is also a really enthusiastic student of science. And she joined me this summer in the research lab to take on an aspect of one of the projects that we have in the lab. And this work was funded by the Burke Endowment that Dr. Pickard helps oversee. So this is a really great example of a student taking on a project and then making it their own and really delving into the parts and pieces. And we have greatly enjoyed having Maki in the lab over the summer and through the academic year, and she keeps surprising me with the Maki things that she does. So, Maki's going to tell you what she did.
Thank you, Dr. Vaughn, for the introduction. And as Dr. Vaughn said, but with a different pronunciation, my name is Macarena. And I'll be talking about the effects of non-introduced sweeteners on lipid accumulation on candidiasis elegans. So first we're going to start defining what are non-introduced sweeteners. Or from now on, I'm going to abbreviate it as NNS. They are non-caloric or low-caloric artificial sweeteners which do not have any nutritional benefits. There are very four that are well known everywhere. The first one is aspartame, which is the active ingredient of the brand Equal. We have acyl-sulfate potassium, which is the active ingredient of the brand Sweet One, and it's the one that's most commonly used in diet drinks. We have sucralose, which is the active ingredient of Splenda, which past research in our lab has been done on sucralose and Splenda. And then last but not least, saccharin, which is the active ingredient of Sweet and Low, and it's the one that I'm going to be talking about due to being the best studied and the most used. Many studies, especially like European-based, have proposed that sweeteners activate sweet-sensing receptors, and this hypothetically leads to food cravings. This causes also metabolic disruption by taking the glucose out of the blood and storing it as fat. NNS consumption has shown evidence of prevalence differentiation into adipocyte cells, which are fat cells. But many of these results remain arguable today. How does this happen? NNS disrupts the homeostasis of the system by messing with the normal functioning of mesenchymal stem cells. This type of cells create cartilage cells and bone cells. When exposed to artificial sweeteners, it differentiates more prevalently to adipocytes, fat cells. This hypothetical finding is especially important in kids, since it could have a very great impact in their growth. Why did we choose this study? Well, this study was motivated by an increase in obesity and type 2 diabetes among the population worldwide. Since 1996, these two diseases have been increasing year by year. Both diseases are right now major public health concerns and are approaching epidemic proportions globally. They're both multifactorial diseases, meaning they have factors that come from genetics and from the environment. These two right here are heat maps, so the hotter the color, the higher the prevalence. We can see that most hot regions coincide in both of these maps, meaning that these diseases are hypothetically linked. One of the environmental factors that have been extremely studied due to causing this disease would be sugar. Sugar is in extremely high proportions in many of our daily foods, even when we don't know. Sugar has been described as empty calories. So what did we do? In order to replace the sugar, we developed NNS. And little by little, they became more popular. And even associations like the American Heart Association and the FDA declared it safe as an alternative for those that were trying to reduce the sugar intake in their diet. But it is notable that the increase in these two diseases correlates with an increase in the use of NNS. In this graph, we can see in the x-axis, years from 1980 to 2013. And we can see how the sugar consumption drops around the year 2000. But in 2002, right here, we can see that the use of artificial sweeteners was 9%. When we approached year 2017, that consumption went up to 41%. As we look at the obesity line, it's been increasing constantly. And even though the sugar consumption dropped, the obesity line kept increasing. As part of why we chose this study, it really called out that the global epidemic is not only occurring in adults, but it's also happening in kids. And these alternatives are now more present in products that are aiming younger generations. In this image, we have kids Pedialyte, and it has two of the NNS that I named before, azasulfame, potassium, and sucralose. Not only in this type of products, but those that are labeled as healthy by the market use NNS constantly, like this great 100% protein yogurt. One of the healthiest cereals in the market, Fiber One, uses sucralose as well. Nowadays, even drinks that are not non-diet drinks use NNS in order to reduce the calories that they contain. In this graph, we can see the youth obesity rates at new highs worldwide. From 1996 to 2016, we can see how the obesity prevalence has been increasing, especially in first world countries, such as the North American or the European continent. But we have a problem. Nutritional studies on humans are not only unreliable, because nobody remembers what they ate at this time 2 weeks ago. They're also inconsistent, because we are all different. And they're also very unethical. If we're trying to see at the effects that these products have, we can't just force someone to take it, especially if we're talking about kids. So for this reason, we chose a model organism, which a model organism is a non-human species that's been extensively studied in order to perform scientific research on it. And for this study, we decided to choose the model organism Planoherditis elegans. So what are C. elegans? C. elegans are free-living nematodes that they live in the soil. We can find them outside, but we just buy them. They have a transparent body, which was very useful for this study because I could look right through them through a microscope. They also have a very rapid life cycle. They go from an embryo to a full-grown adult in 55 hours. They go through 4 larval stages, labeled L1, L2, L3, and L4. We can see that after L1, they could go through this hibernation stage called dower. They would go through dower if they have limited food, dense population, or high temperatures after the L1 stage. Dower is just a stage in where the metabolism slows down. So these nematodes would be characterized by being smaller, thinner, and move slower. These nematodes could be hermaphrodites or males, but only 1% of the whole population are males. It's very convenient that the genome has been fully mapped. And then it's important for our study that 80% of the C. elegans genes are orthologous to human genes, meaning that they're so evolutionary related that they're likely to function in the same way. Do you remember when we learned in biology that everything that has similar structure will have a similar function, and vice versa? The pathway we were most interested about was the insulin signaling pathway. We were lucky because this pathway is very highly conserved from invertebrates to mammals. In C. elegans, it is known as the IGF-1 signaling pathway. And it's very well studied due to its relationship with the dower stage. Its key receptor or regulator is the DAF2, which is known to regulate reproduction, aging, and lipid metabolism independently from one another in this species. And here we have a graph representing the pathway. Up here to the top in red, we have the DAF2 receptor, which is a key receptor, which is going to activate the kinase. And the kinase is going to activate the AKT1, which activates this transcription factor, DAF16 phospho, which is the most important transcription factor that we're going to be looking at. This is going to be located in the nucleus. The transcription factor is going to create 8 different transcripts that encode for genes that influence longevity. But based on the literature read, these 8 transcripts that we were looking at are the ones likely to contribute in changes in fat metabolism. This led us to our research question. Will the exposure to NNS lead to an increase in lipid deposition? I wanted to follow any abnormalities when exposed to the sweeteners, since it's an essential key to this study. Before knowing if the mechanism is being disrupted, we need to see if there's any physiological differences in the nematodes when they're exposed to different concentrations. And I would like to know that this is continuing work from Ashley Reynolds and Nastasia Gilbert, and the three of us did a lot of troubleshooting in order to get a working protocol to quantify the lipid deposition. So I want to talk a little bit about the troubleshooting that we had. We had a hard time developing a protocol that would stain the worm, let us see where the lipid distribution was happening, and would also quantify in an easy way in order to see the data in a graph. We were trying to follow DAF16, that transcription factor I mentioned before. So we used this certain strain of C. elegans, TJ356, but it turns out that the dye was blocking the fluorescent activity of the tag. So whenever we tried to image the nematodes under the microscope, we could not see the fluorescence. We can barely see it in this picture here a little bit, but we could barely see it, as we can see in this one right here. That took us a couple weeks to figure out. And then also, due to having limited resources, we could not look at a gene expression. So finding the most accurate quantification method was the key. I decided to go with all red all dye, which based on the literature, it corresponds to biochemical measure triglyceride man. It is a lipoprotein stain, or a lipophilic dye, which means that it loves lipids. It's going to adhere to every lipid it sees. It is very accurate when representing lipid content based on all the literature read. And it is also a qualitative measurement for lipid distribution. We could perfectly see where the red was in the nematodes. Now we need to know where the lipids are going to distribute in the nematodes, because if not, how would we know if it's abnormal? We know that most of the lipid-dense areas in the nematode are going to be the hypodermis, the intestine, its gonads, and its eggs. Although most of the worms I looked at, they had no eggs, because the ones I looked at were in L4 stage, so we're still in larval stage. For my research, the strain of C. elegans used was the TJ356. And then we used 2 strains of E. coli, OP50 and NA22. On the first day, the nematodes were synchronized, which means we put them on the same life stage. We'll wash a plate and we'll get the eggs from those adult worms. We'll retrieve the eggs and we'll put them in the incubator. That way, on the second day, 18 hours later, those eggs would already be L1 nematodes. And would expose them to what we call treatment day, which we're going to talk about in a little bit. 24 hours after that, we would move these nematodes to plates. The tubes were simply dumped into NGM plates, which are nematode growth media plates. And many of you may wonder, why do we treat the worms in tubes? And it is because that way we ensure that the E. coli was not going to be a co-founder of the experiment. That way we ensure that it was the nematode, the one receiving and absorbing the treatment. And finally, on the 4th day, we performed the all-red-O assay, which an assay is just an examination, analysis. And we're going to talk about it in a little bit too. So as I mentioned, the glucose assay, it was performed on the 2nd day. We separated the nematodes into 5 groups. We had control, which was our negative control. We were not expecting any type of change. We also had glucose, which was our positive control. It was 10 millimolar glucose. We were expecting those nematodes to have an increased lipid deposition. Then we had our sweetener treated groups, 10 millimolar and 30 millimolar. And then 10 millimolar saccharin, which is sweet and low active ingredient. Many may wonder, why did I use the active ingredient as well as the sweetener if it's an active ingredient? Well, many toxicology researchers say that it's important to use the active ingredients, and some of the inactive ingredients could have an impact in it. And then, how did we do this? We'd get a microcentrifuge tube, and we would add 100 microliters of the sugar or the treatment, 5,000 nematodes, and then we would dilute to a total of 1 milliliter. The other assay that was performed was the old red O assay. And we would perform this assay after washing the plates in which the treated worms had been growing for already 24 hours. So in 3 microcentrifuge tubes per group, so if we had five groups, we would choose 3 technical replicates. We would have 3 microcentrifuge tubes per treatment group. We would end up with a total of 15 microcentrifuge tubes. We would put 200 nematodes and then 100 microliters of M9. We would centrifuge, and then it would leave us with a very tiny nematode pellet. 200 nematodes are very, very tiny, so we had to be very careful to not disrupt the pellet every time we would manipulate the tube. We would take the supernatant out, again, being very careful, because you could take the nematode pellet out and not realize, and you would do the whole procedure without treating the worms, which has happened. And then having a nematode pellet, we would add 600 microliters of old red O dye. It was kind of tricky to figure out how much dye to add, so many papers were read, and 600 microliters was the best way proportionate to 200 nematodes. Then we would leave the 15 tubes mutating 2 hours in the dark. It's important to know that all the replicates had to be treated the same way. We had to note any changes in temperature from one biological replicate to another. It was very important to stay consistent. After the 2 hours, we would centrifuge the 15 microcentrifuge tubes. Then we would be left with the nematode pellet, and we would take the dye out. The pellet was again very tiny, but this time it was black, basically black. Then we would add 600 microliters of M9, and basically what we're doing now is washing the extra dye that was not adhered to the nematodes. We would leave the 15 tubes mutating for 30 minutes. This was ensuring that all the extra dye would be out of the pellet. We would take 600 microliters of supernatant out, and then we would shake. After that, the nematodes were ready to be imaged. So we would add 3 5 microliter drops into a slide, and we would take it to the normal microscope. These are some of the results we saw. These are example images, so we can appreciate how control has a way more clear body than any of the treated groups. These little dots are extra dye, because we could perform the wash once, but if you wash it like 5 times, you'd see less of the background notes. We were just trying not to get any dye around them, so they would not influence the red pixel count. In the 10 millimolar glucose group, we can see how the nematode gets a little bit darker, especially in this area, and the area closer to the head. When we see the sweetener-treated groups, they're clearly darker than control. And then this 10 millimolar saccharin group is darker, but then we also saw some nematodes that were kind of darker around the intestine and the gonad, but not as dark in the hypodermis. As we see that the red pixels increase, those are the groups that we were expecting a higher lipid deposition. So those red pixels is what we use to quantify the lipid deposition. Here on the y-axis, we have the red pixel count as percent control. In the x-axis, we have our treatment groups. Control, 10 millimolar glucose, 10 millimolar sweetenol, 30 millimolar sweetenol, and 10 millimolar saccharin. A total of 311 nematodes were examined, which that equaled 5 biological replicates, so the experiment was performed 5 times in the exact same way, with 3 technical replicates per group. Every time, 10 to 15 nematodes were examined per biological replicate. The statistical test used was one-way ANOVA, and what we see is that the sweetener-treated groups showed a statistical significant increase when compared to control, but the glucose-treated group didn't. What we could conclude from this result is that there was a greater increase in lipid accumulation for the sweetener-treated groups, even when compared to the glucose one. There was only significant increase in those treated with the sweetener, but there was not a significant increase in the 10 millimolar glucose group, which we did not expect this. Thanks to not expecting this, we can therefore think that there's either a mechanism disruption or the nematodes are consuming more food. Why do we think that? Linked to the background information before. If these sweeteners are causing food cravings, these nematodes don't have self-control. They're not like us. They're going to consume more food if it's available. The overall conclusion would be that there's a greater lipid accumulation using sweeteners. So why are we using them to help obesity, if obesity is an excessive fat accumulation in the body? We also think that NNS have to be causing some type of disruption in the mechanism, but the mechanism change remains unknown today. And it's true that right now more research is being done with sweeteners in mice. As future studies, quantifying the amount of E. coli consumed by each group of nematodes would be excellent to know if they're actually consuming more food. And thankfully, this is in progress. It would be very nice to see how DAF16 plays a potential role in the lipid metabolism and how its transcripts are encoding different genes that would change what's happening. Then it would be very nice to compare Nile red and oil red. Nile red is another quantification dye. It just doesn't adhere to lipids as well as oil red does based on the literature, but it would be good to have it as backup. And then it would be super convenient to do a comparison between a biochemical assay, which is like a nematode slurry, and then you just quantify the micromolecules, and oil red lipid quantification. I would like to thank the Worm Club team for helping me out this summer. Dr. Wong, Kelly, Alex, Becca. Thank you, Dr. Baker and the Bergen government for trusting me. Nate, thanks for your help. And then my brothers and sisters, since I would not be here without them. These are my works cited. And is there any questions?
Can you go back to your day-by-day experimental design?
Yeah.
So how many days do the worms spend where they really don't have anything to eat?
Well, in the treatment day, they technically have to eat, like they're absorbing the treatment, but it would be like 24 hours. It would be 24 hours without a food source, without E. coli.
And then can worms taste sweet?
I don't know, that's a good question. I could look that up, I actually don't know that.
It's a little bit difficult for me to, when you make that leap of lipid deposition to obesity, right? So a few days after, 2 days after you treat them, you let them rest, let them grow, and then you see greater lipid deposition in the ones that are treated with sweetened or artificial sweeteners. I get that. Did you track all these that showed higher lipid content over the next few days and see that that sudden accumulation of lipid might just be a shock for the worms to actually deal with suddenly eating so much, like your body goes into shock as you suddenly consume a lot of sugar. Like you say, when they have a lot of food, they're going to eat a lot of food. But how many days after that is there possibly some kind of mechanism whereby it's homeostatically brought down back into some, good levels of lipid, rather than it be, because when you say obesity, it's a long-term thing, it's
Yeah.
fixed. So for me, when that sentence is there of making that leap from higher lipid deposition after a few days of treatment to obesity, to me seems a little bit sort of a leap. Do you agree, or did you track them?
After I treat them with the old red, they die. As soon as I add the dye, they die.
It's fixed, then you die?
Yeah.
Okay.
They die. So it would not be possible. What we could do to check that would be more like move them to plates and leave them a greater time in the plate without treating them.
So add the
And
dye
then
a
image
few
them.
days later, rather
Yeah,
than...
imaging them after, instead of 24 hours, wait a little bit longer.
Is that in the plans, or that might be something that
It could be in the
I think
plans.
would be interesting to see,
Might be in the
whether
plans now.
it's just like a sudden reaction, or is it a long-term dealing with it?
Yeah. Well, the dye kills them. So
But I
after,
think
after
your
that,
suggestion is correct, that rather than do it on day 4, you could
I
probably
could-
do it on day
yeah.
5 or day 6 and see if there are any difference in that.
see,
Yeah.
we could do that. We could potentially do that. That would be an easy fix.
Have you thought of a good follow-up experiment to this?
Could you repeat that?
Have you thought of a good follow-up experiment to this?
Yeah, so as part of what Dr. Allen said is actually a very good follow-up experiment, but what we've been thinking is quantify. What we're starting to do now is quantify how much these nematodes eat, because if it's because they're consuming more food, looking at the mechanism disruption might be easier because we're not expecting that big of a change if it's just because they're consuming more food. So a follow-up would be quantifying how much they eat and then getting more data.
So I know like European countries are a lot stricter on what they allow as ingredients.
Yeah.
Are these as prevalent? Like are these sweeteners as prevalent?
They're not. They're not. Actually in here, like whenever you ask for a coffee, they give you sugar and sweeteners. At home, they only give you sugar.